In this lecture, we're going to talk about the molecular basis of inheritance. In other words, what is the molecule made of that is forming these hereditary units that Mendel was working with. And, I mean, this is something that's well known now, but it was a long discovery process. So just to go through the historical perspective briefly, it was back in the 1850s that Mendel developed his basic laws, first and second laws of genetics, which then 30 years later, when they discovered meiosis and mitosis, that led to the chromosomal theory in the 1900s, which said that which put together Mendel's work and the discovery of mitosis and meiosis to come up with the idea that the hereditary units are located on chromosomes. And we talked about how that was first really strongly supported by the work of Thomas Hunt Morgan working with fruit flies. Okay, so notice that it took about 50 years from the chromosomal theory to the determination that it is DNA that is the genetic material. So that's quite a number of years to go from, the idea that the chromosomes contain the genetic material to identifying DNA as the material. So first, let me just mention that chromosomes are made up of a single long molecule of DNA, but they also contain quite a few proteins that are important in helping to package and fold the DNA. So between the 1900s and the 1950s, it was really a more popular theory that to suppose that, or hypothesize that, proteins served as the genetic material on the chromosomes and not DNA. And so we're going to look at some of the classic experiments that led to the discovery that, in fact, it is DNA that is the genetic material. So for a long time, I think scientists thought that DNA was too simple. It's just made out of 4 basic types of building blocks that we'll review or talk about today. It seemed too simple, perhaps, to have this very complex role of carrying the genetic information for all of life on Earth. Proteins, on the other hand, are more complex, more varied, more diverse, and therefore they were thought to be a better candidate for serving as the genetic material. So one of the earliest experiments, and you will be expected, by the way, to recognize these scientists that we're going to be talking about and their contributions. So back in the 1930s or 1928, there was a researcher, Frederick Griffith, who performed a very pivotal experiment where he was working with strains of streptococcus that cause pneumonia. And they knew that there was a pathogenic strain where the surface of the bacteria was smooth, so they called it the S strain for smooth. And then there was another related strain, but that had a rough surface. That was the R strain for rough. And that was found to be non-pathogenic or non-disease causing. Okay, and so what he did was he mixed the pathogenic strain where he had already heat killed the bacteria. So they were dead, the pathogenic strain, but he mixed them with living rough bacterial cells that are non-pathogenic, and he found that some of those living cells actually became pathogenic. So from that he surmised that there was some kind of transforming principle, something that was passed from the dead smooth strain, pathogenic strain, to the harmless living R strain. So what was that transforming principle? That was the question. So here's the experiment that he did. In this first scenario, we see that he took the living pathogenic S smooth cells, injected them in these poor little mice, and the mouse dies. Okay. Then he took the living R cells, the rough cells without the capsule around it, injected the mice, they're okay. Then he killed the S cells, these are bacterial cells, so he heat killed them, injected them in the mice were okay. But then this was the pivotal part of it. Then he mixed the heat killed S cells with the living R cells, injected the mice, and they died. So this is where this idea of the transforming principle came from. Even though the smooth pathogenic cells were dead, something in them was passed to the rough cells to allow them to become pathogenic themselves. Okay, so the question then was, is it something in the chromosomes, the DNA, bacteria? There was a lot of questions going back and forth about which type of organic molecule was serving as the genetic material. And as I was saying, the proteins were the more popular hypothesis. Almost up until about this time, in the early 1950s, when there was this classic experiment done that pretty much settled the case in favor of DNA being the genetic material, which we now know is the case. But this famous experiment was by Hershey and Chase. Okay. And once this experiment was published, it was very difficult for scientists to argue any longer that proteins were the genetic material. So we're going to look at this experiment. You are responsible for it, so make sure you study it and understand what's going on in the experiment and how it's showing that it's DNA and not proteins that is serving as the genetic material. And going back to Griffith's experiment, that the transforming principle that was being passed from the smooths to the rough strep bacteria was DNA. So first of all, before we get into the actual experiment, I want to talk about bacteriophage. These are viruses that infect bacteria. So just as we have viruses that we have to contend with, so do bacteria. Okay, and like in us, viruses inject their DNA, or in some cases, like the coronavirus, RNA, into our cells. That's what viruses do. So that's what they were working with, the Hershey and Chase. So here's a picture showing 3 bacteriophage sitting on a bacterial cell, and notice they're injecting something into the cell, which we now know is DNA. So a virus rather, is primarily made of a protein coat and some kind of nucleic acid inside. Like I said, DNA, or in some cases RNA. And then some viruses also have a membrane, a lipid capsule around them. Anyway, this is what they were working with. So the bacteriophage that Hershey and Chase worked with is primarily made out of like a protein outer shell with some some DNA inside. There's the DNA inside. So the question was, what exactly is being injected into these bacterial cells by the virus? Is it the proteins, or is it the nucleic acid inside? So here's the experiment they did. First of all, it's good to know a little chemistry here to explain this. Proteins contain sulfur, but DNA does not. So they used radioactively labeled sulfur to label the proteins in the viruses. So they grew up viruses in the presence of this radioactive sulfur. Okay, and then they also, in a separate batch of viruses, they grew them or let them replicate in a medium with radioactive phosphorus, which is found in DNA but not in proteins. So each group, in each experimental group, either only the proteins, those are the pink ones, or the DNA, the blue ones down here, were labeled with these radioactive labels. Okay, then they let the phage, or the viruses, infect the cells, the bacterial cells, and they then took the cells and put them in a centrifuge. And if you remember from lab, in a centrifuge, heavier components go to the bottom of the tube and lighter components stay on the top, so you can separate things by their density. So notice that with in the tube, there's going to be some material that's outside the cells, because the virus doesn't completely go into the cell. It injects part of it into the cell, and the rest of it stays outside of the cell. So you have the actual cells, and then you have the material outside of the cells. Okay, and that's true for both experimental groups. So the question was, what is the virus injecting into the bacterium? Is it DNA or is it protein? So they did the experiment, they got their pellets, they spun them, and then they analyzed the results in the end, and they looked in both groups, where is the radioactively labeled material? So up in the protein group, they found that the radioactivity was in the liquid surrounding the cells, so it was not injected in the cells. But down in the DNA experimental group, they found that the radioactively labeled DNA was inside the bacteria. So that is what the virus was injecting into the cell. This is the transforming principle that Griffith was alluding to in his earlier experiments. So this was a very powerful experiment to support the idea that it's DNA and not proteins that is carrying information, the genetic information. And it wasn't long after that, like the next year, when the famous team, Watson and Crick, came up with the three-dimensional model of DNA, the structure of DNA. So they had some help from Rosalind Franklin, who collected the X-ray diffraction data, where she used X-rays to sort of photograph DNA. You get a diffraction pattern where the X-rays are bouncing off various atoms, and based on this is a famous photograph of hers that she took. Notice it has like an X shape here. If you're a trained crystallographer, anyway, I guess this X pattern helped her come up with this idea that DNA, it helped give information to come up with the structure of DNA. And Watson and Crick actually used Rosalind Franklin's data quite a bit. It really informed their model, although she didn't get a whole lot of credit in their famous paper that came out in a few years later. But she is now recognized as part of that, the important scientists that discovered the structure of DNA. So let's look at it. Nucleotides are the building blocks. So we covered this briefly at the very beginning of the semester. So just like amino acids are the building blocks of proteins, or monosaccharides for carbohydrates, nucleotides are the building blocks of DNA, and also the other type of nucleic acid, RNA, that we'll be talking a lot more about in future lectures. So here's a nucleotide in this box here. It has three basic parts. It has a phosphate group, Remember that as one of the functional groups we talked about long ago. You have like amino groups, carboxyl groups, carbonyl groups, phosphate groups. So there's a phosphate group on there. There is a sugar. Pentose means 5 carbons in the sugar, so not like glucose, which has 6. So this, is a building block of carbohydrates, but it also is a component of nucleotides. In DNA, this sugar is called deoxyribose. In fact, that's what the D stands for, deoxyribonucleic acid. So you have this deoxyribose sugar. If you're talking about RNA instead, it's a slightly different sugar called ribose. RNA, ribonucleic acid. So there's the sugar, and then the third component is the nitrogenous base, which means a nitrogen-containing ring-shaped structure, of which there are 4 types in DNA. So let's look at that next. Here we see the 4 different nitrogenous bases rather. So in every nucleotide, the phosphate group, which we see over here, and the sugar are the same. They're not different in different nucleotides. They're the same. But then the nucleotide can be one of these 4 types. Usually they abbreviate them with a single letter. So you've got thymine, it's already labeled, adenine, cytosine, guanine. So you can see if you look at them closely, you see they all have nitrogen in them, ring-shaped structures. So we talk about guanine nucleotides, cytosine nucleotides, adenine nucleotides, and thymine nucleotides. And those are the 4 building blocks of DNA. So again, this is, I think, a large part of the reason why it took scientists a long time to realize that something this simple in terms of its makeup could carry so much information. And we'll see how it does that in a later lecture. So notice also in this picture that these nucleotides are connected together here. So they're connected with each other to form a string or strand of nucleotides. So DNA is actually made of 2 of these, 2 strands of nucleotides that are connected together by hydrogen bonds. And that is not shown in this picture. We'll see that in a moment. Here's another view of the nitrogenous bases. Cytosine and thymine are referred to as pyrimidines, and adenine and guanine as the purines. Uracil is found not in DNA, it's found in RNA instead of thymine. So RNA also has 4 types of nucleotides, except uracil replaces thymine, and it's a different sugar, slightly different sugar, ribose instead of deoxyribose. Okay, we'll talk a lot more about DNA later. Today we're focusing on, on RNA. We're focusing on DNA. One of the benefits of looking at X-ray crystallographic or diffraction data is that this is what gave Rosalind Franklin the idea that in order to fit the diffraction patterns that she saw with DNA, the only way it would really fit with the 2 strands of DNA is to have a purine in one strand juxtaposed to a pyrimidine in the other strand. So that's the only pairing that seemed to fit her crystallographic data, or her diffraction data rather. You couldn't have 2 pyrimidines or 2 purines, it just didn't fit the diffraction pattern. So this was a big clue, and also coupled with something else that had been noticed by somebody named Chargaff. It's called Chargaff's rule. He noticed that if you look at any species, the percentage of adenine in a species was roughly equivalent to the percentage of thymine, and the same with cytosine and guanine. So say in some sea urchin or something, the 38 the percentages of the adenine and the thymine would be similar, and the same with cytosine and guanine. So this is where they came up, or he came up with this idea of the base pair rule, that if you have adenine in one strand, the nucleotide in the other strand that it was pairing up with by hydrogen bonds. Remember, dots represent hydrogen bonds. Would be a thymine. So they call this the base pair rule. Adenine pairs with thymine, and cytosine pairs with guanine. So if you look through here, notice it's A and T, C and G, or G and C. Base pair rule. And also, if you look, one's a pyrimidine, one's a purine. So this is fitting the X-ray diffraction data as well. And this is the structure of DNA, basically. So this picture, I think, is quite informative. Let's look at it a little more carefully here. First of all, notice that the phosphates and the sugars are on the sides like a backbone, and then the hydrogen, or the nitrogen bases rather, stick into the middle of the molecule, where they base pair with each other by hydrogen bonds. So sort of like a ladder. Okay? And it makes sense that these nitrogen bases were sort of in the center of the molecule, in the middle of it, because, and this was something Rosalind Franklin described, because they're hydrophobic, whereas the backbone phosphate and sugar groups are hydrophilic. So DNA is a double-stranded molecule. There's two strands of nucleotides. Two strands. Okay, now something else, it's important you pay attention. This might seem like a minor thing at first, but notice these labels here, 5' end and 3' end. Okay, what does that mean? Well, if you look at the sugar a little more closely, the carbons have numbers on them, and they aren't always drawn either, but there is a carbon at the corners here of this pentose sugar. So this is carbon number 1 here. Here's carbon number 2. Carbon number 3 has this hydroxyl group hanging off of it. Okay, that's the and there's nothing attached to that hydroxyl group, so they call this the 3' end. It has on the number 3 carbon, there's nothing attached to it. If you look at the and continuing to number, this next corner is the 4 carbon, and then here is the number 5 carbon right here. So if you look at the other end, the number 5 carbon, which has the phosphate group attached to it, is there's nothing beyond that. So that's the free end. So we call that the 5' end. So if you have a strand of nucleotides, there's going to be a 3' end, and there's going to be a 5' end. Again, the 3' end has a free hydroxyl group on the number 3 carbon of the sugar. The 5' end has the phosphate group on the number 5 carbon. Now look at the other strand. Notice it's the inverse. The 5' end and the 3' end are flipped, or what they refer to as anti-parallel. They're parallel to each other, but they're reversed. So, anti-parallel, that's what it means. Okay, this is going to come back and be important when we talk a little bit later about how DNA is copied. Okay, and then just to get the final view, a little more correct view of DNA, look over here. We see that this ladder-shaped double-stranded helix, or double strands of nucleotides are twisted into a helix. So here we see the helical nature of DNA, and that's why it's often described as the double-stranded helix. Two strands wound into a helix. Alright, this is just showing the base pairing, and once again, these pink dots are hydrogen bonds. Now remember, because it's been a while since we talked about them, hydrogen bonds are not as strong as covalent bonds in cells, so they're going to be much easier to break. And we'll see later, this is going to be important because you need to be able to separate the strands of your DNA at times. For example, when you want to copy the DNA, you need to do it. And then later when we talk about how DNA contains genetic information, when you want to use that DNA, express the genes, you got to unzip, or you have to break these hydrogen bonds so that the strands can separate temporarily. So it's sort of like you can zip and unzip your DNA by breaking and reforming these hydrogen bonds. This is a space-filling model of DNA. Okay, it's showing the helical nature of it. Okay, now this has a lot of text on it, but again, you make sure you know who Watson and Crick are. They describe the DNA structure. They noted that the structure of DNA gave rise to a very elegant mechanism for copying your genetic material. Because if you know the sequence of one strand, so like let's say one strand is A, D, C, G. So if that's the sequence of one strand, and then I ask you, based on that, does the sequence need to be of the so-called complementary strand? You know, what are these base pairing with? As long as you know the base pair rule, you know that it's going to be thymine, adenine, guanine, cytosine in the other strand, because you've found A and T, C and G rule. So if you think about it, this is really a very elegant way of replicating, which just means copying, the DNA. So their hypothesis was that during replication, the two original DNA strands, which they're calling the parent strands, are going to unzip. The hydrogen bonds are going to break. They're going to come apart, and then the cell will just make a new complementary strand using the old DNA parental strands as a template. So here's a picture of the parent molecule. See, it's got two strands, base pair rule, very simple. You want to copy it, which you need to do during, remember, the S phase of the cell cycle. That's when you would synthesize new copies of DNA. So you first got to separate the two strands. Again, that was by breaking hydrogen bonds. And then there's going to be nucleotides in the cell. You can start assembling a new complementary strand shown in lighter blue, one for each parental strand. So they call the parental strand the template strand, or the parental strand, and then you're making a new complementary strand off of that. So that was in their paper, and partly why their paper is still talked about today, even though it was published back in the 60s, and they received the Nobel Prize for their work. Okay, now we now know that mechanism that we just talked about, how the two parental strands separate and new ones are made, that is now the acceptable model of DNA replication. However, I do want you to know how they supported that hypothesis. So there's another famous experiment we're going to go over that showed that this mechanism of replication is the way DNA replicates. They call it the semi-conservative model because in the two new DNA molecules that are made, notice they have one old strand and one new strand represented by the different colors. So the old is the darker blue, the new is the lighter blue. Okay, and that's what we were just talking about in the previous slides. So I'm just adding now that that's what they call it, the semi-conservative. Semi-conservative because in the two new so-called daughter DNAs, or first replication round, each DNA has one old and one new strand. So the old strand is partly conserved in each one. Okay, but now there was, that initially they didn't know if that was the way it worked, or it could have been the conservative model where the DNA parental strands separate, new complementary strands are made from that, but then when they zip back up, you have the old parental strand zipping back together and then the two new strands zipping together. So you end up with one old and one new DNA molecule. And so we call that the conservative model because the original parental DNA was conserved during replication. All right, and then there was another one to rule out called the dispersive model, where the two new DNA molecules made, where every strand contains some old and some new. Okay, and I don't think this was the most popular hypothesis, but it is something you have to rule out because it could happen that way. So here's the name of the experimenters that carried out what they call the Messelson-Stahl experiment that supports the semi-conservative model. So again, this is another experiment that I encourage you to study it and make sure that the different steps make sense, that you understand it, because you will be asked questions about this on your test. Okay, so here's what they did. They took bacteria cultured in medium with heavy nitrogen, a heavier isotope of nitrogen, and DNA has a lot of nitrogen in it. Okay, and then they transferred that bacteria to medium containing a lighter nitrogen isotope. Okay, so you've got like heavy and light, we'll call it nitrogen. So they knew that the bacteria replicate every 20 minutes. So after the first 20 minutes, they took a sample, took the DNA, and they put it in a centrifuge, and then they looked at where did they see the DNA. Wherever there was DNA in the tube, they would be able to see it, image it. So they looked for how many bands are there in the tube. So in the first round of replication, these pictures down here show you what you would expect to see if that is the correct model. So in the conservative model, remember that means that the original DNA is conserved, and then you get a whole new one. And remember, these have different weights now. The older DNA is heavier, it's got the heavier nitrogen in it, so it's going to move further during centrifugation, further down in the tube. So you would see two bands, so the old and the new band. Whereas in the semi-conservative model, you would just see one band because both new DNA molecules have an old and a new. So they have the same weight, so they're going to migrate the same distance in the tube, and you're just going to see one band. And then the same would be predicted with the dispersive model because the two DNAs that are produced are the same weight. They have some old, some new in them. So they did the experiment, and this was their first result. They saw one band, not two. So that's why there's a big red X through the conservative model. They ruled it out in that first round. but they needed to do a second round to distinguish between semi-conservative and dispersive models. So after 40 minutes, they took another sample, centrifuged it, and they found 2 bands this time. So let's look down here at what you'd expect. For the semi-conservative model, and look back at the first round, look at these pictures, when this DNA molecule right here separates, then there'll be an old and a new DNA molecule, but then the other strand is already new, so there'll be another new strand made, and you'll end up with 2 completely new or light DNAs and 2 heavier ones. And so you would get 2 bands. In the dispersive model, they would still all be the same, so you would predict 1 band, and they saw 2. So the second round eliminated the dispersive model, and that was the support that this experiment gave to the semi-conservative model. Okay, now let's look at how DNA replicates. How do you make copies of it in more detail? I mean, we just talked about in the big picture. The 2 strands separate, and then you use each one as a template to make a new strand using the base pair rule. But let's look at the nitty-gritty steps involved. It's actually a process that involves multiple proteins. Here's the main enzyme, DNA polymerase. DNA polymerase is the, well, there's different versions of it, but this is basically the enzyme that adds nucleotides to the DNA to synthesize it. And notice that the nucleotides have 3 phosphate groups on them initially, so it's a nucleoside triphosphate. And then when the DNA polymerase adds it, the nucleotide, 2 of those phosphates are clipped off, and that's what is supplying the energy for this, because this is an anabolic reaction. Remember when we talked about that? Anabolic reactions require an input of energy. So this triphosphorylation of the nucleoside is what's providing the energy. Just like in an ATP, when you clip off phosphate groups, it can release useful amounts of energy. Okay, so we're going to use the classic bacterial chromosome to look at replication, how DNA is replicated. But bacteria have circular chromosomes, bacteria and archaea. Eukaryotes have linear chromosomes. So we're going to be looking at bacterial DNA, but a lot of what you're going to learn is applicable to linear chromosomes, although there are some differences that we'll get to later. But we're starting with bacterial DNA. So what happens when the cell wants to copy its DNA replication, is that the 2 strands will separate at what they call an origin of replication. And that can happen multiple times. So it's like, you know, the 2 strands are the blue lines. So they come apart, they're separated, that's called a replication bubble. And then the DNA, once the strands are separated, the cell is going to make a new complementary strand in the lighter blue based on the parental template strands. So we're going to look at, we're going to zoom in on one of these areas right here, what they call the replication fork, and look at DNA replication. So the first time you study this might seem quite complicated, because it is, but you know, you might have to go over it a couple times before you feel comfortable with it. So I'm just going to kind of walk you through the main events that happen. Okay, so first of all, you have to separate the 2 strands. In the dark blue in this picture are the original parental strands. The light blue is the new strand being made. Okay, so you have to unzip the molecule, the DNA, and that is done by a protein. Oh, it's not labeled here. Okay, it's called helicase. So it helps kind of unzip the DNA. And then you have these little gray round things here are referred to as single-stranded binding proteins. Single-stranded binding proteins. So they help stabilize the unzipped DNA. Okay, so those are two important proteins involved in replication. All right, and then we have one of the main proteins involved, DNA polymerase 3, it's called. DNA Pol 3 is DNA polymerase 3. Okay, this is the main enzyme that's laying down new nucleotides to form the complementary strands. However, DNA polymerase 3 has some limitations. So you could say it's kind of a picky enzyme. And because of that, it makes replication more complicated. So there's two things that DNA polymerase requires in order to lay down a new nucleotide in the growing DNA chain. First of all, it can't start a strand on its own. It has to have a little piece of RNA to start from. So that we call that a primer. And they're showing that in red. So these little red areas here that you see, the primers, that's actually RNA. RNA I didn't talk about much, but it's single-stranded, not double-stranded, and I did mention a few other differences, but we'll get more into DNA or RNA later. But for now, you have to have a little RNA primer for the DNA polymerase to start from. And then the other thing is that DNA polymerase 3 can only add to the 3' end of a growing chain. It can only add to the 3' end. So that introduces some complications. In one of the two strands, it's not too much of a problem because the cell would lay down a primer, a little piece of primer. Oh, and there's an enzyme for that. Here it is, primase. So you need to know that one too. Primase is what lays down the primer, the RNA primer. So in one strand, you just need one primer, and then the 3' end is free for the DNA polymerase to just start adding nucleotides. And it's going to add nucleotide after nucleotide, and it's going to be growing the chain, and as it does, the DNA is going to keep unzipping, and so it just keeps adding, and they call this the continuous chain, the leading chain, rather. The leading strand, sorry. And it's pretty straightforward. You lay down a primer, and then you start the DNA with primase, and then the DNA polymerase 3 adds to that 3' end of the primer. Sounds confusing. All right, so that's the easier chain. In the other strand, there's a bigger problem because the 3' end is away from the replication fork. The replication fork is in here. So the DNA polymerase 3 has to add in the other direction because you can only grow DNA in a 5' to 3' direction because you can only add at the 3' end. You can't add to the 5' end. Therefore, on the other strand that they call the lagging strand, it's a lot harder for this because you have to make a bunch of little primers. See them, the red thing? You have to make a bunch of little primers, and then the DNA polymerase 3 adds new nucleotides to that primer away from the replication fork until it gets to the next primer. So you're building it in pieces. You've got like primers at intervals, and then pieces of DNA in between. So it's the DNA part is built in fragments. Here's the name of them. They're called Okazaki fragments, named after the scientist who described this. Okazaki fragments. Okay, so you're building the chain, the new complementary strand, away from the replication fork in these little pieces. And then once you do that on both strands, eventually you have to remove the primer. You got to remove, because you don't want RNA in there. So you have to remove that and replace it with DNA nucleotides. And that's the job of DNA polymerase 1. So it's a different polymerase. And it can remove the RNA and replace it with DNA. Okay, but it can't make once it does that, once it replaces the RNA with DNA, it cannot connect to the previous strand that was made. So you need yet another. Let's see if you look at this picture here. The RNA has been completely removed, but there's a little gap there because you need to form a covalent bond here to splice the fragments together. So you need yet another protein called DNA ligase, which knits them together. So that's the lagging strand. And once again, it's complicated mainly because the DNA polymerase 3 can only add to the 3' end of either the primer or the growing nucleotide DNA chain. So this is just a quick run through of all those proteins we talked about. You do need to be familiar with what they do. So you need all of these helicase to unwind the double helix, single-strand binding protein to stabilize it, topoisomerase, I did not mention that before, but that as you unwind the DNA, you also tend to get overwinding ahead of the replication fork, so topoisomerase is correct for that. Then you need the primase to make the RNA primer, the DNA polymerase 3 to make the Okazaki fragments and to elongate the leading strand, the DNA polymerase 1 to remove the primer, and the ligase to knit them together. So those are the replication proteins. Now, the pictures that they show you make it look like they travel along the DNA like a train or something. But actually, it's thought that all these proteins are in like a huge complex called the replisome, and that it's actually the DNA that feeds through it rather than these proteins traveling along the DNA. Okay, so read about that in your book. It's probably DNA replication is probably something you need to go over several times if you get into the nitty-gritty of all the proteins involved. Okay, now bacteria don't have this problem, or cells with circular DNA, but for eukaryotes with linear DNA, linear chromosomes, there isn't a problem with replication. And that is that every time you copy a chromosome during the S phase of the cell cycle, which you're going to do every time you want a cell to undergo mitosis or meiosis, then there's a problem in that there's no way to complete the 5' end once you remove the primer in one of the two strands. So if you look at this middle piece of DNA here, the primer was removed, but now you have a 5' end there. And remember, I told you DNA polymerase III is picky. It can't add to the 5' end. So what ends up happening is that the chromosome actually gets shortened. And the next time there's a round of replication, this is not going to be replicated either. So you end up with shorter and shorter. When they say daughter molecules, they mean, you know, the chromosomes, the replicated chromosomes. So they get shorter and shorter every time the cell divides. So this is thought to have a lot to do with aging and why some cells can only divide X number of times, because they're shortening every time. So some DNA, or well, actually all DNA, all the linear chromosomes have what are called telomeres at the end. Telomeres are pieces of or DNA that's not, they're repetitive sequences of nucleotides that are not carrying important genetic information. In other words, you can do without them in terms of you're not going to lose any important genes. But so it's okay if the telomeres get shortened with repeated replications. That's okay. You're not losing any important information. So while they don't prevent the shortening of the DNA molecule, they do prolong the any adverse effects that might come from having your chromosome shortened. So they sometimes describe telomeres as like the cap on your shoelace, you know, the tip of shoelaces that kind of protects the DNA and from losing important information. Now some cells, not all, but here are the telomeres that are stained. Some cells, not all, but some, have an enzyme called telomerase. And telomerase allows those cells to lengthen their telomeres. So this is important, for example, the stem cells that give rise to germ cells, gametes, egg and sperm, white blood cells, which some of them have to multiply many times during your lifetime. They have this telomerase enzyme. You know, but other cells do not have it. So a cell that has telomerase activity can live a lot longer. I mean, it can replicate a lot longer. And they found that many cancer cells are actually able to do this, to lengthen, to re-lengthen their telomeres so that they can multiply again and again and cause tumors and so on. Okay, so that completes Chapter 16 on DNA structure and on DNA replication.